Hello, and thank you for coming to our presentation. My name is Chris Field, and I am president, co-founder, and principal developer at Theia Scientific. With me is...
Hi, I'm Kevin Field, and I'm a professor and also vice president at Theia Scientific as well.
Today, we're going to talk about three things that do not appear to overlap in any way, shape, or form, but hopefully by the end of this presentation, we will convince you that there is some overlap there. Specifically, we're going to talk about fusion power, Grafana, and machine learning. And what those rule three are going to be doing is being interweaved together with a technology that we call the Theiascope. And so we're going to show how all three things come together. And we're going to start first by talking about fusion energy.
Yeah, so as I'm a professor, we have to start with Fusion Energy 101 and Fusion Materials 101 as part of that. So what I want to talk about is how fusion energy is being used and how it will power AI data centers, but more importantly, how we know they're going to last over the decades in order to power those data centers. And that's where materials becomes important. So under fusion energy, you're going to have a reaction that occurs that makes energy, right? Or basically gives you the energy that you need for a data center. But it's also going to create something called neutrons, right? And those neutrons act as tiny little bullets that are going to make tiny little holes in your materials. And ideally, we want to know if it's going to make the holes and how many and how big. And that's going to be a theme that you're going to hear throughout this. And so we have computer simulations that show us essentially how these materials are going to be hurt. But if you're a regulator in the US or even in the EU, computer simulations isn't enough. We actually have to do experiments. But we have a problem. The problem is that we don't have a full-scale fusion energy center at this time, right? We don't have a tokamak. ITER's still being built in France. And so what do? In my research group, what we end up doing is using ion beams. And so these ion beams you can think of as basically little laser guns that are shooting the same type of holes in the materials that the fusion system would as well. And so we use the Michigan Ion Beam Laboratory in order to irradiate those materials and look at how they're going to perform and try to mimic or simulate what's going to happen in the tokamak. We actually compare that with computer simulations, but we're effectively doing experimental simulations as part of that. And what my group specializes is actually seeing these defects or this damage that's created into the material, seeing them in real time. So we actually connect a microscope into the ion beam facility, and we use a microscope called a transmission electron microscope. For those that are old enough, it's like an overhead transparency projector with with electrons instead of white light. So basically, you magnify the image using the microscope, but you're also bringing in that laser beam of ions in order to hit the material. And then we end up being able to see the damage that's made in real time. So each one of these little black dots and loops is actually damage in the material. And what we want to know is how many and how big we have. And we want to know that over time because we want to know the dynamics, because we want to know if we're going to have this material last for seconds, minutes, decades, so on and so forth as part of that. This is what it actually looks like in real life. This is one of my graduate students operating the microscope. So you have the microscope here, and that ion beam comes in, and we are able to view everything digitally right on the microscope. For reference, this microscope is $2 million as part of it. And so it's a very fancy piece of equipment as part of that. And so what the students will do is they'll take videos and they'll take images on that very fancy microscope while we hit the sample. And then they want to do is figure out how many and how big all of those little black dots are in the image as part of that. And then eventually we have a discovery, and hopefully at some point I'll be up here on a stage with a Nobel Prize, right? But we have a problem. So if you're a microscopist, this is our beautiful dashboard. So this is the software that most people are using. It's called ImageJ. It is basically Windows 95 grade type of material, and it's been used for the past 3 decades. And so if I submitted this, I don't think I'm ever going to get a Golden Grot in this conference using this software as part of it. And you can see how bad this is. The other problem is that all of that is done with manual labor. My students sit there and they manually circle every single one of those little black dots that I showed in the video or I showed in the image. And so that's where AI and ML also gets interwoven. So what we've been doing over the past couple years is starting to use the same things for self-driving cars and so on and so forth in order to start doing automated defect detection in our images. And the scientists have actually gotten pretty good at this, right? So we've gotten really good at using real data as well as synthetic data in order to train the machine learning models to find the same features in the images. And that has rapidly accelerated our ability to look at data, right? And they keep on getting improved every day or every week. And there's quite a few groups now that are actually developing these types of algorithms to do that automated type of defect detection. For those that are machine learning practitioners, the F1 scores in our field is kind of around 0.8, and it keeps on growing up to 1. That's like a B- student for those that use the US grading system as part of that. But there's another problem. Scientists are really good at making these machine learning models because they just take them off the internet, the open source models and stuff like that, and use them and train them for it. But they're really bad programmers at deployment, right? So almost all of them are using Colab or Jupyter Notebook with built-in dependencies and so on and so forth. So if I wanted to use another research group's data source, it ends up being almost impossible, right? We have this problem where they have specific dependencies, there's poor formatting, and there's poor UI or no user interface as part of it. And the other problem becomes, my group, I want to do this all in real time. I want to see what's actually happening on the microscope. And so until a couple of years ago, there was no solution for this. Right? And so what Chris is going to show and talk about is our web app and our hardware platform that allows us to do all of those things together.
Yep, thank you. So what we've come up with is this technology that we call the Theiascope, right? And this is combining Grafana and Jupyter notebooks all on a device that you can see here on the left-hand side. So it is an on-premise self-hosted device that is attached to that microscope because we don't really want our multi-million-dollar microscopes being accessible to the outside world. So we brought the GPUs and we brought the cloud to the microscope in this case. Now, I could probably talk about this diagram all day on this sort of thing, but I think it would be better and easier just to show it in action. So let's go ahead and we will hold our breath and hope this works, and we'll flip over to the system. So this is our home dashboard. This is the central interface, the first interface that scientists will see. And we can see that we have a couple different panels. We're using a calendar app panel that Kevin really enjoys because he can tell whether his students are actually doing work or not, and if they're in the lab or not, based off of when data appears. On the other side, the left-hand side, we have our model management panel. This is where we can spin up a variety of different models. I've already marked a favorite for today. And we're going to look at some TRISO particles, and I'll explain what those are in just a moment. We're going to, again, hold our breath, and we're going to click the Start Actor button. This brings out a drawer that has all those machine learning model parameters that allows you to tune and fine-tune the inference that's occurring on the system while you're running your microscope. But we're just going to use the defaults, and we're going to hit Start. And this is going to unpack that model, load it onto the GPU that is sitting right here, hopefully, and it will then spin up. So it's initializing it, it's unpacking it, it's getting it ready and spun up to do it. We're now idle and we're ready to go, and we can do some experiments. Now, we didn't bring our $2 million transmission electron microscope with us. They wouldn't let us have it through security. So instead, we're going to use some images that we'll use in sort of approximation for that. And what I'm going to do is now we've moved into our acquisition panel. And here's where we're going to be able to capture some images. And we are going to do some real-time image analysis on these TRISO particles. So these TRISO particles are, if you're familiar with the US candy, the gobstoppers, there are sort of inner layers of them. Right? And what we want to know is the uniformity of each of these particles. And we've collected, in this case, we have about 10 images, but in reality, you probably have thousands, if not tens of thousands of images of these particles. And normally, you would have to draw the circle around each layer and then measure those layers and then decide, is this a circular particle or not? If it's not circular, we want to reject that batch of particles. So we have that set up. I'm going to move this field of view so that we can say that we want to look at just the part that is in the center here and not grab the rest of the stuff. It takes me just a moment to do this with the trackpad. And we're going to go ahead and we are going to start some acquisition. So it's going to go in progress, and in just a moment, with luck, on the right-hand side, we started our very first inference, and we're running through those images. Now, I'm going to sort of say, okay, let's move around and let's see and look at some other particles while we're doing this, right? And this is just going through preview on the Apple. We can see in the other side there that we are clicking through, and it's doing that analysis in real time. So we just have to use our imagination a little bit that this would be on the microscope as it's being connected to his ion beam, where they're simulating a fusion power system to power the next generation of data centers that it's ultimately going to literally and figuratively power Grafana in the future, right? So we're going to go through and do a couple different ones as we sort of scan through these images. This is just to give us some preloading for some other things that we're going to see here in just a moment. So I've loaded up those. We can see this one's a little oblong. We can see that as humans, but what we can also do is, well, we can zoom in, but we can scroll down, and I have this uniformity chart. And this uniformity chart is if it was a perfectly circular particle, each one of those layers would be a perfectly flat line across the plot. So this allows the user, this allows the scientist to immediately see, hey, is this a good particle or a bad particle? But we also have on the other side some stat panels that show the standard deviation. And so we can turn those into metrics that will identify then and aggregate if this is a good sample or a bad sample in all of this. On the left-hand side, we have our different layers. This is just sort of the performance of the model, how confident it is it was able to find those layers, and we can go through the system and look at that. And so what we can also do is we can zoom in, we can do some further analysis, and we can do that further analysis by going to what we call the examine dashboard. And now the examine dashboard, if this also hopefully works, will give us the same kind of information. But for those of us that are old enough, this is like our VCR. You can scroll through in time, you can go back in images, and you can go forward in images like we were doing it during the acquisition. That panel doesn't seem to want to be working. It's a little shy today, so that's fine. We'll go back over to our acquisition panel. And what we can see here is we still have this uniformity chart. And what I'm going to do is we'll go in and we'll take a look at what's going on. How are we calculating these uniformity plots? And what we're doing actually is we're running something that doesn't look like PromQL or a SQL or something. We're running this notebook. We're running a Jupyter notebook, right? And how are we doing that? Well, in fact, we have Jupyter running on the system, and every time the dashboard updates, we are running through this Jupyter notebook that a graduate student wrote to create that uniformity plot. This is then in a language that the scientists are at least familiar with, and they can download these and they can upload them to the system, but it's running this code every single time within this panel, giving us this uniformity. And this is what allows the scientists to create whatever kind of metrics, whatever kind of plots they want to have during the experiments while they're on the microscope. But we can also do some other things with this, right? Because the way Jupyter is working and the way it's hosted on the system is, yes, we can do it with notebooks, and we can also use dashboard variables. We can pass those to the notebooks. But because of the way it's working, we can actually do this, and we can put, as an example, we can put, if I can scroll again, the Python code directly into the dashboard. So we are running this live on the system, and we can make some changes. If I don't like the parabola, we can go ahead and we can make, if I could type, we can go ahead and make that change, and we can do that as we are doing the experiment. And we can copy and paste snippets of code from other notebooks or from other researchers or from other collaborators, and we can make these changes in real time while we're running the experiment. And like I said, we also had this tied to various variables, or we can create these other variables in the system as we're running them. Right? So we can do these things in all of this different way, and we're able to create these custom code snippets and run the code live inside the dashboard in an environment that the scientists are familiar with, given that they're developing these models that they want to host and deploy at the microscope to ultimately make their discovery, determine if it's a good material or bad material for a fusion power plant, and or win the Nobel Prize. Right? That's the dream. So we go ahead and I'm going to stop the acquisition, and we will go back now to the presentation. Great. So what we saw there is the whole thing working together in real time on the microscope at this point. So the question is going to be, how did we put this together, and where do we go with this from here? Right? And this is going to be the very first question, which we always get, which is, okay, why'd you do this with Grafana? Why'd you pick it? And we picked it for a variety of very good reasons, right? One is the data sources, where there's a whole community of them. They're open, they're available. That also helps with this big tent thing, because that's the interoperability that we have that allows us to connect to a variety of different microscopes and experiments and customize our dashboards. The very first iteration of this entire code and application that I showed Kevin was, the very first feature request was, I want a dashboard where I can change things and move things around and have them for different things. And I was like, okay, we're going to go with Grafana because I don't want to have to create all of that. And we get all of this for free, in a sense, by doing that. And the other nice thing is that we have some great panels and visualizations that we can use, the documentation for creating your own visualizations and data sources and plugins is fantastic, and that has really helped us to develop this. And then also there's this vibrant ecosystem of all the people here and around the world putting this all together for us and helping us out in doing this. Also, Grafana is awesome, right? We're all here, it's awesome, but is it rad? I'm going back to the 1980s, right? But rad in the sense of, can it be used to do irradiated materials characterization? And also, can it be used as a rapid application development environment? Well, we already have seen something that is somewhat similar in that we have an interactive, notebook-centric development environment that's pretty ubiquitous among the ML and the science community that rapidly ingests, explores, and visualizes code and or data. Well, that's interesting because that's also what Grafana and the observability stack does. So in actuality, though, spoiler alert, yes, we can make Grafana rad because we just saw the demo doing that, and we wouldn't be standing up here if we couldn't do that, right? But how did we get to making it rad? And it starts with the anatomy of a panel. So I sat down one day and I was thinking like, okay, how does this all get put together? How does this all work in a sense? And if you look at this and squint at it, you can sort of see the backwards G of the Grafana in there. In the flow, you go from your data source to the query editor, you're creating data frames, and you're going to use the transforms. Then you have your options, and they ultimately get put up into the view inside of a panel. So using that, what we have really is that your query is your selecting data. The transform is converting it into a format that can then be customized by the options, and that can then ultimately be displayed inside the view. And if we focus in on those first two, what we have is we have our query, and then we have our scripts, and those are transforms and the snippets, respectively. And those where the code is. But I'm sorry to say, scientists are never going to learn SQL. It is just not something that they can wrap their brains around. They're too busy operating those microscopes and drawing boxes around black dots. So it's not Python. That's what they're learning, that's what they use, that's what all the notebooks are in. So what else is out there that is small snippets of code that does some kind of translation of maybe a request, pulls some data from a database, and returns a response? And that's what got me thinking. And I was like, well, maybe we could use something like Lambda from AWS, which runs Python and JavaScript. But can we do that on the device? Can we have a similar kind of thing running small snippets of Python code in an environment that's familiar to scientists and engineers? And yes, we can. And that's what this Jupyter flow we're calling here. And this is what was running on the demo. You start off with your Jupyter server. We had our Jupyter lab, which is the web interface for that. But we've created a data source that passes Python code from Jupyter lab into the server, and then comes back out. But we can also go the other direction. We can go from our query editor, which we saw with the inline code, and we can pass that back to the server. This allows us to create some pretty crazy things to do with the kernels and the various Jupyter notebooks running, and we can do things like pass the dashboard variables between them. But we're entering the Jupyter flow into and integrating it into the Grafana flow of that panel, right? We're going from the query editor to the transforms, options, and view. But what this makes it happen is the Jupyter notebook becomes the data source, right? It becomes that part that is translating it from one to the other. And we can do some, like I said, some pretty interesting and kind of crazy things. We can run the code directly in there. The scientists can make the changes. They can see it happen immediately. They get that interactivity and that feedback in the system. But they also then have the ability to install the entire Python ecosystem, because we are also handling the magic keys with the % sign there. So we're doing a pip install here just of time zones, because we needed to do that in order to do the sign there. But we also now provide a rapid application development environment for scientists and engineers in nuclear materials characterization or irradiated materials characterization, right? So Grafana is rad. It is awesome. It is rad. And we're able to create these custom workflows and have them run on a deployable system that's easy for scientists and engineers to use. And then also share, ultimately, all this information to get us to fusion energy to power the next generation of data centers. So what we hopefully have done is convince everyone here that these three seemingly disparate things are actually interwoven together, that we have also done that by using, and we have our Golden Grot there because we were the 2024 Golden Grot winner of the beautiful dashboard, and that dashboard was generated by using this exact system. So there's the sort of plug-in for that situation. But in the center is this Theiascope that is connecting the fusion energy and the AI ML together. And on top of all of that is the Grafana stack integrated with a Jupyter Notebook interface. So with that, we'd like to acknowledge and thank some of our collaborators and our funding agencies, specifically the Department of Energy in the United States. But the images that you saw of the TRISO particles came from Oak Ridge National Lab. Some of the models that we use and everything come from Wisconsin. And then Idaho National Lab and Argonne National Lab are both collaborators that have let us deploy the system in their multi-million-dollar microscopes in a very early stage of the system and device. And with that, thank you.
Thank you.